Next story. MIT University's silicon spikes eliminate 96% of virus particles. In a groundbreaking study led by MIT University, an innovative virus-killing surface has been developed, boasting an impressive 96% efficiency in eliminating virus particles. This cutting-edge technology features a surface made of silicon adorned with minuscule nanospikes that effectively skewer viruses upon contact. The research, showcased on the cover of the prestigious journal ACS Nano, demonstrated the surface's ability to dismantle the HPIV3 virus responsible for bronchitis, pneumonia, and croup. Astonishingly, 96% of the viruses were either torn apart or rendered incapable of replication, a significant breakthrough in controlling disease transmission in high-risk environments like hospitals and laboratories. Dr. Natalie Borg, the corresponding author of the study, highlighted the intricate technical expertise required to design a surface capable of obliterating viruses. Despite appearing as a flat black mirror, the silicon surface possesses tiny spikes specifically engineered to combat viruses. This innovative material could be integrated into commonly touched surfaces, reducing the need for traditional disinfectants and curbing viral spread effectively. The nano-spiked surfaces, fabricated at the Melbourne Centre for Nanofabrication, feature needles that are a mere 2 nanometers thick and 290 nanometers high, inspired by nature's mechanism of insect wings that pierce bacteria and fungi. The research team, led by RMIT's distinguished professor Elena Ivanova, combined theoretical and practical analyses to unveil the surface's efficacy in damaging the virus's structure and membranes, incapacitating a significant portion of viruses within hours of contact. Samson Ma, the study's first author, emphasized the practical implications of this revolutionary technology in enhancing safety measures in high-risk environments, such as healthcare facilities and laboratories. By implementing these advancements, researchers and healthcare professionals can work in safer environments, mitigating the risks associated with exposure to hazardous biological materials. This groundbreaking research was a result of a collaborative effort involving researchers from MIT University, URV, Spain, CSIRO, Swinburne University, Monash University, and the Kaiteki Institute, Japan. Supported by the ARC Research Hub for Australian Steel Manufacturing and the ARC Industrial Transformational Training Centre in Surface Engineering for Advanced Materials, this interdisciplinary study has paved the way for a new era in virus control and containment. The publication, Piercing of the Human Parainfluenza Virus by Nanostructured Surfaces in ACS Nano, marks a significant milestone in the fight against infectious diseases, offering a glimmer of hope for a safer and healthier future. Source: Euric Alert. Next story. Uncovering the origins of racial disparities in childhood cancer through ancient DNA. In a groundbreaking discovery, researchers have unveiled the genetic variant responsible for the increased risk of B-cell acute lymphoblastic leukemia, or, in Hispanic, Latino children, shedding light on the racial, ethnic disparity in this childhood cancer. The study, led by Vijay Sankaran and his team at the Dana-Farber, Boston Children's Cancer and Blood Disorders Center, pinpointed a mutation that hampers the expression of the IKZF1 gene, crucial for B-cell development. This mutation, prevalent in Hispanic, Latino kids, was found to be scarce in white non-Hispanic, Latino children, offering a key insight into the disparity. Through genome-wide association studies, GWAS, the researchers delved into populations to identify this risk factor, showcasing the mutation's link to indigenous American ancestral origins. By analyzing ancient DNA samples, the team traced the mutation back to the first migrants who entered the Americas around 13,000 years ago. The study uncovered that this variant, propelled by natural selection, could have conferred protection against infections due to its impact on immune function. Sankaran's team's findings not only fill a crucial gap in understanding the disparities in all risk but also provide a stepping stone towards comprehending treatment resistance and relapse rates in Hispanic, Latino children. By bridging the past with present health challenges, the study aims to pave the way for tailored prevention strategies that could potentially thwart leukemia in individuals carrying this identified genetic variant. This cutting-edge research, published in Cell Genomics, was made possible through funding from prestigious institutions like the National Institutes of Health, NIH, the New York Stem Cell Foundation, the Leukemia and Lymphoma Society, and the Dana-Farber Cancer Institute. The implications of this study stretch far and wide, emphasizing the significance of genetic diversity and ancestral origins in unraveling complex health disparities and paving the way towards precision medicine approaches. With each revelation, science inches closer towards unlocking the mysteries that underlie diseases, offering hope for a future where tailored interventions based on genetic insights could revolutionize healthcare. As we navigate the intersection of genetics, history, and modern health challenges, the quest for knowledge continues to illuminate new paths in the quest for healthier tomorrows. Source: Eradicate Alert. Next story. Impact of severe hurricanes on coral reef sponge population dynamics and genetic diversity. In a groundbreaking study conducted by researchers from Florida Atlantic University, an extraordinary discovery has been made regarding the resilience of coral reef sponge populations in the aftermath of severe hurricanes. These catastrophic storms, Hurricanes Irma and Maria, which struck in rapid succession in 2017, offered a unique opportunity to investigate the recolonization of Aplazina coliformis, a rope-like coral reef sponge with essential importance for marine ecosystems. Before the hurricanes, the A. coliformis population exhibited high levels of clonality, a phenomenon contributing to inbreeding. However, post-hurricanes, the researchers observed a remarkable shift towards increased sexual reproduction for recolonization, with 85% of sexual reproduction stemming from local larval recruitment. This transition not only enhanced genetic diversity but also facilitated the species' ability to combat inbreeding, ultimately bolstering the resilience of Caribbean sponge communities to extreme weather events. The study's findings, published in the journal Molecular Ecology, highlighted that major storm events favored sponge larval recruitment without a significant increase in genetic diversity. The research team, led by Dr. Andrea Chavez Fonegra, demonstrated that the A. coliformis sponge's ability to reproduce both sexually and asexually through fragmentation played a vital role in maximizing dispersal efficiency and successful recolonization of habitats following storms. Through genetic analyses using a cutting-edge genotyping method, the researchers detected larval recruits and fragments at all sampling locations post-hurricane, indicating a potential for rapid population recovery. Surprisingly, the recolonization of A. coliformis after the hurricanes was predominantly driven by sexual reproduction, with gene flow detected across distances up to 60 kilometers between study sites. As our climate continues to change, leading to more frequent intense hurricanes, the impact of such extreme weather events on coral reef species and their ecological interactions becomes increasingly crucial. This study sheds light on the intricate mechanisms that drive the resilience of marine populations in the face of natural disturbances, emphasizing the importance of understanding and preserving biodiversity in our oceans. With their pioneering research, the team from Florida Atlantic University has not only unraveled the mysteries of sponge recolonization post-hurricanes but has also paved the way for future studies on the adaptive responses of coral reef ecosystems to environmental challenges. Their work underscores the resilience and adaptability of marine species in the face of adversity, offering valuable insights into the dynamics of our planet's underwater world. Source: Eradicate Alert. Next story. Enhancing wildlife accessibility. Insights from Virginia Tech study. In a groundbreaking study conducted by the College of Natural Resources and Environment at Virginia Tech, researchers Emily Sinkula and Ashley Dyer have delved into the realm of wildlife recreation and accessibility, focusing on the inclusion of individuals with disabilities. Their findings, published in the prestigious journal Human Dimensions and Wildlife, shed light on the barriers faced by birders with disabilities and offer insights into how to enhance inclusivity in wildlife-related activities. Sinkula, a PhD student leading the study, highlighted the novel approach of merging disability studies with wildlife recreation, a connection rarely explored in previous research. By conducting a nationwide survey of U.S. wildlife viewers, the researchers compared the challenges encountered by birders with and without disabilities. Their aim was to understand the unique needs of disabled individuals in the realm of wildlife observation and conservation. The study revealed that 1 in 3 birders faces accessibility obstacles, such as lack of accessible features, safety concerns, and overcrowding at birding sites. Despite these challenges, both disabled and non-disabled birders expressed a shared desire for access to high-quality birding locations and information on wildlife viewing opportunities. This crucial overlap suggests that efforts to enhance accessibility for individuals with disabilities can benefit all wildlife enthusiasts. Dyer's Human Dimensions Lab, in collaboration with the U.S. Fish and Wildlife Service Multi-State Conservation Grant Program, aims to expand research on wildlife viewers with disabilities. By advocating for inclusivity in conservation efforts and wildlife recreation, the lab seeks to amplify the voices of all individuals, ensuring that no one is marginalized in their pursuit of nature-related activities. The researchers emphasize the societal responsibility to support the participation of disabled birders, urging agencies and organizations to reflect on ways to enhance accessibility and provide necessary training to staff and volunteers. As the number of Americans with disabilities continues to rise, addressing the underrepresentation of this population in wildlife-related recreation becomes increasingly vital. Furthermore, the study underlines the broader societal benefits of including people with disabilities in recreational activities. Beyond fostering social inclusion, enhancing accessibility in wildlife recreation can contribute to mental well-being, strengthen social connections, and promote conservation efforts. By extending the benefits of birding to individuals with disabilities, this research paves the way for a more inclusive and diverse wildlife community. As we navigate towards a more inclusive future, the findings of this study serve as a beacon of progress in bridging the gap between disability studies and wildlife recreation. By amplifying the voices of all wildlife enthusiasts, we can create a more accessible and enriching environment for individuals of diverse abilities to connect with nature and engage in the wonders of the natural world. Source: Eradicate Alert. Next story. Triceratops discovery confirms Spielberg's inspiration. In a thrilling paleontological discovery reminiscent of Hollywood blockbusters, a team from the Naturalist Biodiversity Center embarked on a quest for Tyrannosaurus rex in Wyoming, only to stumble upon a treasure trove of Triceratops fossils. This unexpected turn of events unfolded into a decade-long excavation that unearthed a staggering 1,200 bones and bone fragments from at least 5 Triceratops individuals. Led by the intrepid paleontologist Jimmy DeRoyer, the team delved deep into the mysteries surrounding the Triceratops bone bed. Through meticulous analysis, they uncovered compelling insights into the lives and deaths of these magnificent creatures. Contrary to popular belief, the research revealed that Triceratops exhibited slow growth patterns, shedding new light on the developmental biology. The bone bed itself provided tantalizing clues about the fate of these dinosaurs, hinting at a collective demise, possibly in a swampy setting. Remarkably, the layer of rock encapsulating the Triceratops remains contained no traces of other species, underscoring the unique bond shared by these ancient giants. Delving further into the enigma, the team delved into the physical and chemical properties of the Triceratops' teeth, unveiling a tale of migration shared by all 5 dinosaurs. This groundbreaking revelation hinted at a social behavior that saw these majestic creatures team up, prompting a cascade of inquiries into the complexity of their interactions. Professor Anne Schulp, DeRoyer's mentor at Naturalis and Utrecht University, lauded the monumental findings that have catapulted the institutions into the spotlight of paleontological research. With the largest Triceratops discovery in the world and the first Dr. Triceratops in the Netherlands, the culmination of DeRoyer's work has not only spawned research papers but also birthed an exhibition showcasing the ancient camaraderie of these colossal herbivores. As the world eagerly awaits the exhibition's debut in October at Naturalis before embarking on a global tour, spectators will witness a vivid depiction of the Triceratops herd as they roamed and perished together 67 million years ago. The saga of the Triceratops team-up has not only rewritten the narrative of dinosaur behavior but has also opened a gateway to a trove of captivating new inquiries into the social dynamics of these iconic creatures. In a groundbreaking fusion of science, history, and spectacle, the story of the Triceratops team-up stands as a testament to the enduring allure of paleontology and the unyielding spirit of discovery that drives humanity to unravel the mysteries of the past. Source: Eradicate Alert. Next story. Patients at University of California, San Francisco open to receiving flu vaccination. In a groundbreaking study led by the University of California, San Francisco, researchers have unveiled a simple yet effective method to boost flu vaccination rates among underserved populations, particularly those who rely on emergency departments for primary care. Picture this: patients stepping into the chaotic hustle of the emergency room, not expecting to leave with a flu shot in their arm. But that's precisely what happened when these patients were asked a straightforward question: would you be willing to accept the influenza vaccine? The results were astonishing. Just by posing this question, along with providing informative videos and print materials, researchers witnessed a significant 32% increase in flu vaccine uptake. However, when patients were not only asked but also received a pamphlet, watched a 3-minute video featuring a physician from a similar ethnic background discussing the vaccine, and learned about its benefits, the uptake soared to a remarkable 41%. Flu, a seemingly innocuous illness for some, can have devastating consequences, claiming tens of thousands of lives annually in the United States. Yet, vaccination rates among underserved populations, including the homeless, uninsured, and immigrants, remain alarmingly low. These individuals often face obstacles such as general vaccine hesitancy and limited access to preventive healthcare services. Lead author, Dr. Robert M. Rodriguez, emphasized the importance of addressing health disparities prevalent in emergency departments. The study's messaging approach, including personalized videos and flyers, aimed to bridge the gap in vaccine accessibility and empower patients to make informed decisions about their health. Spanning across 5 cities, including San Francisco and Philadelphia, the study engaged nearly 800 patients with diverse backgrounds. The findings were especially promising among Black and Latino participants, as well as those without health insurance or primary care providers. This systematic approach not only boosted flu vaccine uptake but also shed light on the potential to deliver vital public health interventions to vulnerable populations. Dr. Rodriguez, backed by a team of dedicated researchers, has been at the forefront of advocating for equitable healthcare delivery, as evidenced by the previous success in promoting COVID-19 vaccination in emergency department settings. This study not only highlights the impact of a tailored messaging strategy but also underscores the pivotal role emergency departments can play in promoting preventive health measures. By leveraging simple yet effective communication tactics, healthcare providers have the power to transform patient outcomes and contribute to a healthier, more resilient community. The findings of this study pave the way for a more inclusive and proactive approach to public health interventions, ensuring that no patient slips through the cracks when it comes to safeguarding against infectious diseases like the flu. Source: Eradicate Alert. Next story. Mapping the impact of climate change on wine regions worldwide. In a world where the tendrils of climate change are snaking through every aspect of our lives, perhaps one of the most unexpected victims is the humble grapevine. From the rolling hills of Bordeaux to the sun-kissed valleys of California, wine growers are facing a new and daunting challenge: how to adapt to a rapidly changing climate that threatens to upend centuries of tradition. A team of dedicated researchers from the National Research Institute for Agriculture, Food, and Environment, INRAE, has been meticulously studying the effects of climate change on wine-growing regions around the world. Their findings paint a vivid picture of a landscape in flux, where once thriving vineyards are now at risk of withering under the relentless onslaught of rising temperatures and extreme weather events. As the mercury climbs higher and higher, vineyards in traditional wine-growing regions such as Spain, Italy, and California are finding themselves on the front lines of a battle for survival. The delicate balance of temperature, rainfall, and humidity that once sustained bountiful harvests is being irrevocably altered, pushing many regions to the brink of collapse. If global warming exceeds 2 degrees Celsius, the very heartlands of winemaking could be rendered barren, their grapes too parched to produce the nectar that has sustained generations. But all is not lost. As some regions falter, others are rising to meet the challenge. Northern France, the Pacific Northwest, and even unexpected locales like Belgium and Denmark are finding themselves newly hospitable to the grapevine, their cooler climates now offering sanctuary to a plant in peril. By embracing new techniques and resilient grape varieties, these regions are not just surviving, but thriving in the face of adversity. Yet, the road ahead is not without its perils. The specter of new diseases and pests looms large, threatening to undo the hard-won gains of proactive growers. Adaptation is the watchword of the day, with strategies ranging from soil preservation to vineyard density adjustments being deployed in a bid to stave off disaster. The very essence of wine, its taste, aroma, and character, hangs in the balance, a delicate dance between tradition and innovation. In the midst of this turmoil, one thing is clear: the future of wine is at a crossroads. The decisions we make today will echo through the vineyards of tomorrow, shaping the very essence of what we pour into our glasses. As the world warms and the grapevines tremble, it falls to us to ensure that the spirit of this ancient craft endures, a testament to human ingenuity and resilience in the face of a changing world. Source: Eradicate Alert. Next story. Unlocking genetic variant behind childhood leukemia disparities. USC study. In a groundbreaking study conducted by the Keck School of Medicine of USC, researchers have identified a genetic variant that sheds light on the heightened risk of acute lymphoblastic leukemia, or, in Hispanic, Latino children. This particular variant, found on the IKZF1 gene, has been pinpointed as a key contributor to the 30 to 40% increased likelihood of developing all in Hispanic, Latino children compared to non-Hispanic white children. Through genetic fine mapping analysis, the team uncovered a variant that increases the risk of all by 1.4 times, prevalent in individuals of Hispanic, Latino descent. This discovery marks a significant step towards understanding the biological underpinnings of the disease and the disparities observed in different ethnic groups. The research, recently published in the journal Cell Genomics, not only unravels the genetic basis of all risk but also provides crucial insights into the development of screening tools and therapies for this prevalent childhood cancer. Led by Adam De Smith, Ph.D., and Charleston Chang, Ph.D., the study delved into how the variant on the IKZF1 gene influences the development of B cells, a type of white blood cell disrupted in all. By analyzing genetic data from diverse racial and ethnic groups, the researchers identified 3 SNPs associated with higher all incidence, with one variant in particular significantly elevating the risk in Hispanic, Latino children. Further experiments conducted in collaboration with Vijay Sankaran, MD, PhD, revealed that the risk variant impairs the expression of the IKZF1 gene, leading to disruptions in B cell development and potentially increasing the chances of leukemia progression. These findings not only enhance our understanding of the genetic mechanisms underlying all but also pave the way for future advancements in screening and treatment strategies. Moving forward, the research team aims to delve deeper into the implications of the identified variant on patient outcomes and explore its role in the increased risk of all among Hispanic, Latino adolescents and young adults. By unraveling the genetic complexities of ALL, this study offers a promising avenue for the development of tailored approaches to screening, treatment, and ultimately, improving outcomes for children at risk of this devastating disease. The implications of this research extend far beyond the laboratory, offering hope for a future where precision medicine and targeted interventions can mitigate the disparities in childhood leukemia risk. With continued research and collaboration, the scientific community is one step closer to unraveling the mysteries of all and developing effective strategies to combat this prevalent childhood cancer. Source: Uric Alert. Next story. Empowering Personal Growth, How Deaf Women Excel. In a groundbreaking study conducted by Amaya Jauregui Orbe at the University of the Basque Country, the well-being of deaf individuals was closely examined, shedding light on significant factors that influence their psychosocial development. The deaf community, characterized by its diversity in age, gender, types of deafness, and modes of communication, presents a complex and multifaceted landscape for researchers to explore. One of the most striking findings of the study was the revelation that women within the deaf community exhibit higher levels of personal growth compared to men. Contrary to initial hypotheses, this discovery hints at the presence of empowerment tools within the associative fabric of the deaf community that enable women to enhance their capacities and autonomy. This unexpected result not only challenges existing perceptions but also underscores the importance of considering gender dynamics in understanding well-being among deaf individuals. Moreover, age emerged as a significant factor influencing psychosocial well-being within the deaf community. Individuals under the age of 30 were found to have higher self-esteem, greater perceived social support, and increased personal growth compared to older age groups. The impact of inclusive education on this age disparity was highlighted, suggesting that those who received inclusive schooling may exhibit higher levels of well-being. Furthermore, the study delved into the correlation between educational background and well-being among deaf individuals. Prelingually deaf individuals educated in mainstream schools demonstrated higher self-esteem than those who attended special schools, pointing to the potential benefits of inclusive educational environments. Additionally, the level of education achieved was linked to higher levels of self-esteem, personal growth, and social well-being, with those holding university degrees showing superior well-being outcomes. While linguistic, cultural, and occupational factors did not directly influence the psychosocial well-being of deaf individuals in the study, the researcher emphasized the importance of exploring the intersectionality of various variables for a comprehensive understanding of well-being among the deaf community. By analyzing the complex interplay of factors, future research endeavors may uncover nuanced insights into the factors shaping the well-being of deaf individuals. With a robust research design and inclusive methodology, the study incorporated the perspectives of 166 deaf adults representing diverse backgrounds, ensuring a comprehensive examination of the psychosocial well-being of the deaf community. By adapting assessment instruments to accommodate the varied needs of participants, the research aimed to provide a nuanced understanding of the factors influencing well-being among deaf individuals. Amaya Jauregui Orb's pioneering work not only contributes significantly to the scientific literature on the well-being of deaf individuals but also underscores the importance of considering diverse factors in understanding psychosocial development within the deaf community. As the research continues to evolve, the study paves the way for further exploration and insights into the intricate dynamics of well-being among deaf individuals. Source: Euric Alert. Next story. Australian soil emissions fuel global warming. Curtin University study. In the vast expanse of Australia's soil lies a ticking time bomb, a potential future contributor to global warming. The once trusted carbon sink is facing a troubling fate as the Earth's temperature rises. Recent research conducted by a global team, led by the illustrious Professor Raphael Vizcarra-Rossell from Curtin University, has unveiled a startling revelation. Australian soil is on track to become a net emitter of carbon dioxide, CO2, by the end of the century. Soil, a crucial ally in the fight against climate change, has been diligently storing carbon, thus aiding in keeping our planet cool. However, as temperatures soar, this beneficial relationship is threatened. The warmer climate is diminishing the soil's capacity to retain carbon, leading to potential carbon release back into the atmosphere. The research team, through meticulous simulations, has projected the fate of Australia's soil under different societal scenarios, from an eco-focused sustainable pathway to a bleak fossil fuel-dependent future. The outlook is grim. By 2045, under moderate and fossil fuel scenarios, Australian soil could contribute more than 14% of the country's total emissions. A concerning prediction considering the detrimental impact on global warming. While certain fertile farmlands may continue to sequester carbon, it is not sufficient to offset the carbon loss in more climate-sensitive areas like coastal regions and expansive rangelands, where 70% of Australia's soil carbon is stored. Professor Vizcarra-Rossell issues a stark warning: unless significant advancements are made in farming practices to bolster soil carbon storage, the situation will exacerbate. The repercussions could be dire, with the soil releasing even more carbon, intensifying climate change effects. To avert this crisis, innovative solutions are imperative. Sustainable pathways, enhanced soil management, and conservation efforts are vital for Australia to meet its emission reduction targets. The focus should be on preserving the carbon stocks in rangeland soils through interdisciplinary strategies, innovative grazing practices, cultural burning, and promoting native plant biodiversity. The onus is on us to act swiftly and decisively to safeguard our environment for future generations. The time for change is now. The time to heal our soil and mitigate the looming threat of soil emissions is upon us. The research findings, shedding light on the impending challenges posed by Australia's soil, have been published in the prestigious journal, NPJ Climate and Atmospheric Science, underscoring the urgency of the situation. Source: Euric Alert.